So yeah, it's great to be here. So I'm a postdoc in Fred Chang Lab. This work is a collaboration with Ricky Garner, a student in Julie Theriault at UW. And so I'm going to talk about the cytoplasm. So here are 4 representations from David Goodsell at the Scripps about what the inside of cells might look like. From Luca on the top left to E. coli, pancreatic mammalian cells, and plant cells. And you see that they all have things in common, such as a membrane on the outside to separate inside from outside. And in the eukaryotes, you can see a cytoskeleton element and filaments and things like that. And here a cell wall in the plant cells, or the extracellular matrix here in mammalian cells. But one thing that they all have in common that might not be as obvious is that all of their cytoplasm are extremely densely packed. And so you can see protein in very close contact with other proteins. And I like to think about it as something that defines life, because you have to accumulate things inside of a bag, usually a membrane, and accumulates enzymes and their substrate for reaction rate. And so then when you do that to such a point, you end up with physical consequences, because the internal medium becomes crowded and viscous. And that's what I'm interested about here in terms of biology is how does the physical constraint that you reach inside the cell, how does that affect biology? And so here on that slide, it's 2 published works from 5 and almost 12 years ago now, looking at the physical aspect of the cytoplasm. And so on the left, it's the E. coli cytoplasm reconstructed in that molecular dynamic aspect with protein diffusing around. And what I want you to appreciate is how much contact there is in between proteins and enzymes of all sorts in that cytoplasm. And so you could imagine how that will constrain biology and diffusion rates and things like that. And on the video on the right, it's a more recent work where they also added the 3D structure of the proteins, and you can see how the structure of the protein fluctuates when they come in close contact to each other, and all those frictions. And so that's what we are interested about in that work. And the approach we took is particle tracking. And here we use the GEMS particle developed by Liam Holt lab in New York. And so they are way larger than proteins, around 40 nanometer in diameter and extremely bright. And they are genetically encoded, so we can control the expression level, and we can express them in a bunch of different organisms. And we image them in turf microscopy. And here it's a video to just show you what it looks like when you express them at a relatively high level. You have a bunch of particles in your cell, and you can easily track them and measure their diffusion rate. For the work I'm talking about today, we express them a little lower than that to have a very good accuracy on our tracking in the particles. And so here, just a short slide with the kind of experimental statistics we have here. We have looked at around 150 cells. We have thousands of trajectory with around 30 trajectory per cells. And so we can measure step size for all of those trajectories. And here it's a distribution of the trajectory length in terms of how long we can track the trajectories. And usually the first approach and the first step you take when you have those kind of data is to do a mean square displacement plot. And here I'm showing you what I would refer to as the population level aggregate. So the mean square displacement plot of all of the trajectories coming from all of the cells from all of the day of experiments. And if you do that, you obtain that kind of relatively good-looking MSD plot where for the first hundreds of milliseconds here, you have a relatively linear slope on your MSD plot, and so you can feed that to obtain a diffusion rate and an anomalous exponent. Here, the anomalous exponent is 0.92, very close to 1, so we are going to treat it as a almost perfectly diffusing system. And we can also do velocity autocorrelation on your trajectories that can tell you if you see any viscoelastic behavior. And we never saw the velocity autocorrelation have the kind of negative peak that would be a signature of a viscoelastic environment. But that's just the population level aggregate of the data. And what we got interested about is that if you look at single trajectories, and here I'm showing 5 of the longest trajectories we had in the data set, and if you color code each time point with the step size as a function of how different it is from the average step size, you can see that there is steps that are way longer than the average step size and steps that are way shorter. And so there is a sort of large heterogeneity in the system. And here in that trajectory here, you can see that there is a bunch of small steps all clustered together, and then it makes long steps again. So we got interested in the heterogeneity in our data. And to look at that, instead of doing a population-level aggregate MSD, we looked at it at either what we call the track-wise level, so fitting each of the MSD individually, or then averaging the MSDs that come from the same cells, and looking at the cellular average MSDs. And if we do that, you can see all the heterogeneity we have at the track-wise level, or even at the cell level. Even if we average the tracks that come from the same cell, we have a lot of heterogeneity. And here it's the distribution for the diffusion coefficients that we obtain in the data. And so we have order of magnitudes variability in diffusion inside cells and also between cells. And so here it's another representation of the data. And the first way to analyze that is to do ANOVA test to see where the heterogeneity come from. And so we were pleased to see that most of the heterogeneity did not come from experimental variability, so not from day to day or from microscopy session to microscopy session, but most of it came from the track level. So inside cells, different tracks being different from each other, and also for a large amount also from cells to cells, so different cells being different from each other. And so we really wanted to know, okay, how can we explain that level of heterogeneity with order of magnitudes difference between cellular averages in diffusion and multiple order of magnitudes differences between trajectories from the same cells. And so we took a simulation approach to try to create simulations that would recreate the data and keep it as simple as possible. What is the simplest simulation we need to put together to explain the level of heterogeneity we see in the data? And here we called it the Doppelgänger simulation strategy, because we had the experimental data at the cell level. So for every cell in the experiments, I know their size, I know how many trajectory I have, and for how long I tracked each of the trajectory. So we built Brownian dynamic simulation of diffusion, but we constrained the model to the experimental data statistics, meaning that we have cell 1 in the model and a doppelganger of cell 1 in the simulation that is exactly the same size, that has exactly the same number of trajectories tracked for the exact same number of time points. And that way, the underlying statistics of the simulation are an exact match to the data, and we can easily compare them. And then, if we treat them as being purely diffusive, and you think about Stokenshtein, the only parameter that we have left to play with in the simulation is the viscosity. And so that's exactly what we did. And so we built 4 different flavors of the model. One where what we call uniform viscosity, where the cells are all the same, and inside the cells are uniform, and the viscosity is the same everywhere. Then we had model 2 with spatial heterogeneity, where the cellular average viscosity are the same from cell to cell, but it's broken down in a random pattern inside each of the cells. Then model 3 is cellular heterogeneity, where the cellular average are different, but the inside of each cell is homogeneous. And then model 4, it's a combination of 2 or 3. The averages of cells are different, and also inside the cells are broken down in a random pattern of viscosity. And so if we do that, we can run the model, generate the simulated data sets, and compare their statistics to the experiments. And so here on those plots, the blue are the experimental data here for the tracks on the left and the cells on the right, and the 4 different flavors of the simulation. And so you can see that model 1, the uniform viscosity, we have track-to-track variability and some cell-to-cell variability, and that's to be expected. That's the statistical sampling. Some trajectories are long, some trajectories are short, some cells have more trajectories than others, so we have some level of heterogeneity coming from there, but nothing that compares to the experimental data. And in fact, we can quantify how similar the noise levels are by doing the ANOVA and comparing the amount of variability from the ANOVA to the experiment. And the uniform model only recapitulates half of the track-to-track variability that we see of the experiment, and only 10% of the cell-to-cell variability. But then, as we complexify the model and add the different level of heterogeneity I just presented, so the spatial heterogeneity model, we increase the track-to-track variability and the cell-to-cell variability a little bit, but still not to the level of the experiment. The cellular heterogeneity, we can increase the cell-to-cell heterogeneity, and it can match the experiment, but the track-to-track is not yet matching the experiment. And only the combination of 2 and 3, where every cell has a different average that is broken down in a random pattern inside of the cell, can recapitulate both the track-to-track level of heterogeneity and the cell-to-cell level of heterogeneity. And so even if all of the model can recapitulate the average diffusion rate, only the model 4 can recapitulate the spread we see in the data. which led us to this sort of view of the cytoplasm where viscosity is varying in space as you move through the cell, and where every cell also have a different average. And so the cytoplasm might not be a well-mixed environment at the scales we looked at, and for the time scales we looked at. And then being also experimentalists, we wanted to know if we could get a handle on the level of heterogeneity we have in the data. Is there knobs that we can play with? And so the first thing we did was cytoskeleton or temperature shocks, so removing cytoskeleton, both actin and microtubules. And we didn't see any change either on the average diffusivity or the amount of spread, which, knowing that we work in fission yeast, might not be extremely surprising. There is no actin cortex and things like that. Temperature decreased the average, decreasing temperature decreased the average viscosity, diffusivity we had in the data, but didn't affect the level of noise. And then we did osmotic shocks, so increasing the concentration of the cells by removing water, by adding osmolyte on the outside, and that decreased the average diffusivity, but also increased the noise in the data. So it seems that the concentration of the cytoplasm might be a critical parameter influencing the level of noise in the data. And so that work was a collaboration between me in the Chang lab and Ricky and Julie Theriault, and it originated in the Physical Biology of the Cell course in Woodsall in 2019. And with that, thank you.